I'm Mikko Vähäsöyrinki, so I originally founded Sensapex, who's organizing this webinar. From longer history, I'm also a neuroscientist, and I used to like very much developing new technologies for electrophysiology, so this is quite close to my heart also from that perspective. We have today two speakers here. I will introduce them very shortly together. There will be two sections of this webinar. So the first one is a quick 5 minutes by Marco on tapered fibers in general. And then there's the main presentation by Endre about the experimental side of the things, which is about 25 minutes. After that, we'll have a Q&A session. So during the presentations, if you have any questions, please write them in the chat window. And when the Q&A session starts, we can then start going through them from the chat. And all of you are now muted and don't have a camera on. But once we get to the Q&A session, I will then start enabling also your microphone and the camera so that we can see you if you have a discussion during the Q&A also. If you have anything during the presentations that you want to share, maybe write it in a chat or raise the hand. So I will be in the background and I can check that and see how we do about that. I think that's about the basics. Now let's introduce our speakers today. So we'll have first Marco Pisannello, and Marco started originally studying the telecommunications engineering, and after that he joined the Center for Biomolecular Nanotechnologies at the Italian Institute of Technology, IIT, as a PhD student in 2014. During his studies, he also did 1 year in Bernardo Sabatini lab in the Harvard. He later got back to the IIT as a postdoc in 2017. And during his time in the IIT, he worked on the development and implementation of the tapered optical fibers as optogenetic probes, including their manufacturing aspect. Then in 2020, Marco joined Optogenetics, and he's currently a CTO of the company, and he's been doing the instrumentation development, for example, in Optogenetics. Then Endre Marossi studied originally physiology and neuroscience at Laurent University, doing some experiments also in the Experiment Medicine Institute, with focus on the synaptic and circuit physiology in the hippocampal networks. After that, he became a PhD student at the Cognition and Emotion Research Group led by Sanja Bauer-Mikulovic at the Leibniz Institute for Neuroscience. And there he has been establishing combined optical and electrical interrogation of in vivo circuits, and that's what he will be then talking today. So I think without further words, I would like to hand over to Marco, who can start with the tapered fiber introduction. So I will mute myself. So Marco, please go ahead.
Thanks, Mikko, for your kind introduction. So thanks, Mikko, and I will just briefly introduce the tapered fibers for the audience. It will be a very brief presentation, and I'll be happy to take a few questions at the end of everything. So why tapered fibers? Let's imagine we are interested in investigating a bigger brain structure like the striatum, which is kind of in the millimeters, several millimeter size. When using the classical flat cleaved optical fibers, the volume of tissue interaction is kind of restricted to a size of the fiber core diameter, so in all 3 dimensions, so basically hundreds of micrometer, maybe 400 micrometers for the bigger fiber. So it's quite a limited amount of volume compared to the bigger structure, which can be a feature if the scientist is interested in pinpointing a very localized area. Otherwise, if the interest is in a more broad response from the structure, the mismatch in size between the probed volume, and the whole structure of interest is quite evident. This is why we think tapered fiber can be an interesting tool for optogenetics experiments, because they allow to extend the interaction volume along the fiber of the axis up to several millimeters. Why is that? So you have to imagine that with the classical flat-cleaved fibers, all the light patterns guided within the fiber, all the modes, are emitted at the end of the waveguide where the fiber is cleaved. The interesting point here is that a guided mode, which means a certain light pattern traveling throughout the waveguide, can exist only if the fiber has a big enough diameter. With higher order modes, which are the one with more, let's say, notch and valley in the profile, existing only for higher fiber diameter, while lower order modes are supported also by a smaller diameter of the fiber. If you combine these with the idea of a taper, basically you have that all the modes can reach the tapers, and then the taper itself will select the modes. With higher order modes that reach at a certain point a diameter that doesn't allow anymore for their propagation, and they are pushed out from the fiber, and so exit from the fiber side, while lower order modes can go farther down the waveguide and reach the tip position. And modes in between will undergo the same fate, just at an intermediate diameter. So you can think really of two different operation modality for the tapered fiber. One on the left in this slide, where you excite the full numerical aperture sustained by the fiber, so all the modes are propagated to the taper and the whole taper is illuminated. And depending on how you manufacture the taper, you can obtain an illuminated active optical section from 0.5 millimeter up to 2.5–3 millimeter. On the other hand, if you use a strategy to select a subset of modes into the fiber, which is simply if you scan a laser beam, the angle of a laser beam on the input facet of the fiber, you can select a sub-portion of the taper, either in a random access way, or as you can see in the video on the right, just scanning from the tip back to the end of the taper. And again, this can happen depending on the manufacture of the taper in a total region of 1 millimeter up to 2 millimeter of taper. These are images in a fluorescent solution. I just want to show you also what happened in tissue. So this is a 2 millimeters tapered fiber inserted into a coronal slice, into the striatum of a coronal slice, stained with some fluorescent. And as you can see, we were able to illuminate pretty much the whole extent of the area of interest. And the same fiber was also used with the modal selection, with the spatial selective operation, and we scanned the light emission along the fiber, and interact with different sub-portion of the whole structure. That's basically it. It's the very basic meaning behind the use of tapered fiber. So going on an extended grain structure, interacting with several millimeters of tissue, both all at once, if you like to do that, by injecting the full numerical aperture into the fiber, or subportioning the region of interest by scanning the laser light and then having a smaller light region going up and down to the taper and sub-probing your region of interest. That's basically it. I don't want to take out more focus from Andres' talk, so thank you for this, for your attention in this brief 5 minutes.
Thank you, Marco. So I think we'll then hand over to Endre for his presentation.
I also welcome everyone, and I actually want to express my gratitude that it's a very great honor for me to present what I did with the tapered fiber, and can feel that I'm a little bit of part of this experience. So, and also not only for the organizers, but I would also express my acknowledgement for the team who are also working with me on the results that you will see, my supervisor and our collaborators and our architect, and also the whole group. So what I want to express today is the strategy that I followed at the end for my experiments. I wanted to target the deep brain area, the medial septum, and first, the canonical way to do that is to have a relatively thick optic fiber implanted to the given area, and with my acute experiments immersing neuropixel probe, it would give a very, very low chance of optotagging cells, because you can calculate everything properly, but still in that deep tissue, 1 or 2 degrees of angles are also very much distorting the aiming. And actually, here you just see an experiment where we couldn't find any optotag cells, only cells that were responsive afterward, after the optogenetic stimulation. So we wanted to go for other strategies. In another experiment here, for example, you see 10 seconds of the experiment. That is, I think it's breaking down, right? So it's like 2 seconds, and you see that upon the optogenetic excitation that you see also with the artifacts, only 1 or 2 channels got action potential firing, meaning that we again had a very low yield of optotag cells and reach cells in this area, even if I scroll up or down during these recordings. Only this purple channel was very nice to respond, which of course I was very happy about, but in the long run you cannot do an animal for only 1 cell. So we needed to use another strategy that is actually practical for the demand of the anatomy, as Marco also suggested. So our deep brain area, the medial septum, is actually just having a longitudinal shape that could be very useful for us to use the tapered fiber in these investigations. And the equation is actually quite simple. So I had the probe, I had the fiber, and I got an optrode from it. I just glued here the optogenics tapered fiber together with the neuropixel probe. You see that it has this 10 millimeter long shank, and we discussed with the optogenics guys when I was planning these experiments to use 2.5 millimeter tapered fiber, active length tapered fiber, which resulted in much better results. So 2.5 millimeter means that in our channels we could see optogenetic excitation up to channel number 250, but the whole medial septum was not bigger than 2 millimeters. So for our purposes, it was amazing and very useful. And you can see here in the heat map how the optogenetic excitation is changing the LFP throughout the channels. But you can also see here that we could see a photovoltaic artifact in channel number 225, maybe a little bit in 300, but we didn't see any more up. So it means that the whole idea worked, and we could use it for our analysis. But what did we do? So the question was, what is the role of medial septal glutamatergic cells other than locomotion? How can we grab on them? And for that, I would like to introduce a little bit what is running. So during running, we need to process the environment with a much higher demand. And to make it possible, there should be synchronized a lot of behavioral variables. For example, the whisking in the rodents are very much in synchrony with the running speed. What's more, even have a predictive nature of the whisker movement you see here in gray, but then it gets synchronized with the running speed. Other modalities, actually, for example, the pupil size. So you just really need a better readout during locomotion. And also the subsequent neuromodulation is also known that is orchestrated during locomotion, and also the stepping. And regarding the stepping, the medial septum is nowadays getting more and more known about its stepping signal. So that you see that the animal's stepping, which is marked here, are very much in synchrony with the medial septal unit activity. But it is still not known whether these modalities of locomotion are orchestrated by a single cell type, or it's something that should be synchronized throughout brain areas with recruiting different cells. But what we know about these glutamatergic cells in the medial septum is actually that they are also able to elicit hippocampal theta rhythm. This is a brain oscillation that is very necessary for the function of the hippocampus during navigation. And here the authors proved this with this experiment, where they photoexcited the glutamatergic cells with theta related frequencies, 6 to 12 hertz. And they observed in another brain area in the hippocampus the regarding entrained theta oscillation, and also the speed of the animal was in line with the stimulation frequency. What else did they say is that if they used only a very, very short protocol, like not very short, but like really just 1 second compared to the 20-second protocol, they saw that the induction of theta rhythm already happening during the stimulation phase, and it keep maintained until the locomotion itself happens. So there will be a part in the behavior that we don't really know what is happening there and what is happening with the cells underlying. So we asked 2 questions. One is whether this theta related modulation is really necessary to induce theta, or non-theta related modulation is also enough. So you just basically boost up these glutamatergic cells, and then they will do the job. And the other question is whether they are coding anything else, expect the locomotion initiation, namely whisker movement and pupil is represented by this network or not. So to do that, we use this tapered fiber, and we use the 2 protocols that you also saw before: a long protocol at theta frequency, and the 1 second continuous wave light beam protocol, and the combined tapered fiber that you see if we paint the neuropixel probe with dye eye. Then you see that really this 2 millimeter of tissue is having a very good yield of units, and because our tapered fiber is also in the proximity, because they are moving together, we can yield a very high amount of glutamatergic cell activation. So just to show how it looks in the experiment, here's an animal just sitting on the treadmill during the acute experiment, and around here the laser will turn on. You also see it in the behavior that the animals start running. And during these periods, we monitored the camera motion energy of the face camera to infer the whisking signal, and also applied pupillometry to see the pupil signal. So what we saw, just to repeat the already existing literature's experiment, we saw that we could initiate the running with this long 8 Hz protocol, and we could also initiate the running with this shorter continuous wave protocol. The problem was actually with the success rate, because while this protocol has a very high success rate to initiate running, this protocol is around chance level to initiate the running, and the duration of the initiated run was not different from the GFP control, which is a control that has no channel rhodopsin, but the very same viral construct, only expressing GFP. But what else we saw is that if we take these trials when the animal didn't start run or did start running, and we check the other behavior variables, we see that the CME, that is the whisking, I will refer to it as CME from now on, the CME and the pupil was initiated very reliably, but not the running. And also that there is a sequence of this behavior, that we initiate first the CME, that is followed by the pupil, and later on comes the running. If we check this CME signal more carefully, we also see that there is a predictive manner of the amplitude and the latency of these emitted signals that can infer whether the animal was starting running after the CME happened or didn't start run after the CME happened. You also see here with the color code. So yes, why did we use the neuropixels if we don't have any spiking data? We have. So here you see that during running, actually, some of the units are upregulated or downregulated in this experiment. And if we take the whole the grand average of run initiations, so here's the 0 where the run onset happened. We could identify those cells that were upregulated during running, and also those ones that were a little bit downregulated. But interestingly, this upregulation started already almost a second before before the actual run started. So we were curious whether the coding rather the CME. So we took these micro events when the animal just moved its face briefly, and we saw that actually the very same cells are coding the whisker movement, the CME, when the animal is stationary. So the animal is not running in this sense. And also we also saw that after these micro attentive events, we saw some microdilation of the pupil as well. So I have to hurry up, I think, a little bit, but then we identified a novel cell type that is showing a percent active phenotype, that whether when we had the 10 stimulations, we saw some cells that were having a prolonged, longer lasting activity than the stimulation itself. If you compare it with the cell that is said as non-percent firing cell, you see that usually their activity stops at the end of the stimulation or before, a little bit after. but these cells are maintaining their prolonged activity. And this prolonged activity is actually having a wide range of shorter and also a little bit of longer activity. So if we compare the length of this percent firing to the run durations, we identify that these percent firing cells are coding either the running or the whisking. Note here the different scale. And in total, only 1/4 of these cells are coding something. The 3/4 of these cells, we don't know what they are coding for. But why we are here to also talk a little bit about the optotagging and its success rate. So we used a 1 millisecond light pulse 420 times in order to get reliable optotag results. And you see that here the black dots are actually the arriving action potentials that are very much like synchronized after this 1 millisecond light pulse. This cell is definitely a nice, this is just an example unit. It's a nice example to show. If we take the whole population, we could yield almost 200 optotag cells with a particular latency 1 millisecond after the offset of the optotagging stimulation. And it yielded around 18% of the whole neural population, which is very much in line with the literature. Because by the literature, as the estimate is around 23% of the medial septal cells being glutamatergic. So having a yield of 17%, we were much more satisfied than having one cell during a session. And of course, with the previous approach, you could also have no clue whether your experiment was actually working. You were just in the wrong place. With this, you can be quite certain because you move the optrode. And we also saw that these optotag cells were actually percent firing sometimes with a certain ratio. So from all of the percent firing cells, we could see quite some different cell types. This I would not go into the details because I think it's not belonging to this topic so much. But what I just wanted to show here is actually if we take only the percent firing units, you see that the very same units during successful run induction are firing longer, while during non-successful run induction firing shorter. And finally, I would just talk a little bit about the pupil. If we excited the glutamatergic cells, we saw an increase in the pupil diameter that was not happening in the GFP control. And we saw also that during running, there is a plateau-like pupil signal. Here it is the green. But during stationary episode, when the animal has a micro event of attention inferred from the a micro event of whisker movement, had a peak-like a pupil change that was actually corresponding to some of the cells in the medial septum. So we wanted to see the lagged correlation of these cells to the pupil. And of course, those ones with the run had a broader and higher correlation coefficients because it's a plateau-like signal. But those ones with no run also showing a little bit nicer results. So you see that these almost half of the total amount of the recorded cells throughout these 9 animals were actually very much correlated with the pupil, and actually later on it turned out that actually they are correlated with the CME, and there should be a secondary pathway that concerns the pupil. If we check the proportion of the cells, we saw that out of all units, around 35-40% of the cells were tuned to the pupil. But if we subset only to the percent firing cells, it can reach up to 55, 60, or in some animals, 80% of these percent firing cells were pupil related. So with that, I would like to summarize our findings. So we could use 1 second optogenetic stimulation to initiate locomotion, although with varied success rate. We also saw a percent firing phenotype that we further characterized in vitro, which is also was out of the scope of this talk. So maybe you can ask questions later on that if you are interested. And also that the length of the percent firing was correlated with the locomotion and the pupil. And I just would like to drop here 2 ideas for outlook that you could potentially use. This technique is you can actually combine this technique with fiber photometry, in my mind at least. I'm not sure about the artifacts and so on, but in principle it should work. That could be used, for example, tracking axonal inputs during electrophysiology recordings, which is for my knowledge, it was not really frequently done. And of course, as Marco also mentioned, you can use the tapered fiber to a spatially restricted excitation that could also open new venues and new questions. And I guess also the multicolor registration should be possible, but this I have no idea. Maybe it's also a future outlook. And I would like to thank all of you the attention, and I'm open for questions.
Thank you. So I think we have some questions here, so maybe we start from the first one. This is maybe for Marco. So there's a question that maybe can you read the chat window yourself, by the way?
Yeah, sure.
Yeah. So maybe you can take the first one on the linearity of the tapering.
Yeah, so thank William for your question. So how are the tapered fibers made in order to arrive to a linear shape, reducing hyperbolic shapes? And how do you obtain a very small final tip diameter? So we use a laser puller to do an hit and pull process. And we just optimize all the parameters of the recipe. So the pull velocity, the laser power. There is actually a proceeding of a conference published in 2015. I'm replying to the question in the chat window with a link to the paper. So everyone can access it, and there is all the details you need to know about the tapering process published on that paper.
Cool. Thank you. So then the next one.
Yeah, the next one I think is for me. So it's the neuropixels plus the tapered fiber combination. How far is this from the probe? So like in the contact points of the probe, you should give I think around like a few micrometer more because the contact probe, like the electrodes are facing that direction and the light is coming from behind. But the distance between the tapered fiber and the probe is between 200 to 300 micrometer to have a good photo excitation. The NHP, I am not that familiar what is to replicate in
Maybe I can take that one, but there's a follow-up from the next question about the artifacts. Did you see the artifacts from the optostimulations, and did you stimulate briefly, or how did you manage them?
Yeah, maybe for that I would share back the screen, because I also prepared just 2 examples. So this is a software that we use for the manual curation because, of course, the photovoltaic artifact can be also detected as a spike. So that's an issue. In our experiments, we have this 8 Hz protocol. So we have a clue if we see an action potential that is a noise, that it is indeed a noise. So here you see a normal action potential. You see the footprint of this action potential is varied anatomically. So like the longer, the further distance from the probe will result in a different shape of the action potential. And also that the interspike intervals are relatively nicely distributed. Also, firing rates are nicely distributed, and the amplitudes are very much in the same range. On the contrary, if you go with an artifact that actually the kilosort is very nicely distinguishing, but you can also have it sometimes in a cluster that you should cure it out from. So for that, these spikes are much more unitary. Like you see almost no difference. Maybe this one is the one that is a positive going spike, but by shape it also looks very similar. And as I mentioned, we had the 8 Hz protocol, so we know very well that these peaks are coming from our excitation that you can also see here, and also these unitary responses here. So if I compare the two, it's not only that the spike shape is just so ugly that I could never assign it as a unit, but also other features are helping to filter this out. What the people said, I think from OpenFIS, is that they could reduce a little bit this photovoltaic artifact if they shine the light on the electrode side of the probe. But this I couldn't do because of the holder position. So maybe Sensapex would have a good advantage on that if they have something like a holder to combine this.
Yeah, maybe I pick up that one. So we talked about this at the FENS meeting of the summer, and quite a few labs have made something themselves to get the fiber and the probe aligned for the experiments. But we've been now designing together with Marco and optogenetics a holder or adapter to do it. So I'll just say the screen. So this is an earlier prototype version, but basically here's a cross section where the thing below in a gray is cross sections of our NeuroPixels holder that would hold the NeuroPixel probe with the dovetail cap. And then these are our standard items that are used by quite many labs. And then we modified so that we made the V-shaped groove at the end of it. And the idea is that then you can put the tapered fiber to that V-shaped groove, glue it there to that area, and then the fiber will route back below the NeuroPixel and its dovetail cap, through the way where you can then connect to the fiber, the ferrule there. Now with the normal 1.0 probes, the shanks are 10 millimeter long. So with this one, we can get the fibers roughly 6 millimeter out from the tip. So basically, you have like 6 millimeter depth in maximum you can insert. That requires that the standard dovetail cap is modified, because that's the first place where the fiber will start to collide, because it has certain minimum bending radius that you can't exceed. So that sort of is a constraint for the overall geometry. So without having a cut out in the dovetail, standard dovetail, you get about 3 millimeter, or 3.5, like in this picture, how far away you can get with the fiber. But we are planning to show this at the SFN. So come to our booth if you are there. So we have prototypes there to show how these adapters look the neuropixels with the fibers on them. With 2.0, there's a different adapter that we have, and we are going to have a version for that also. Only one fiber, even if it's 4-shank probe. With the longer ones, that there was a question here for the primate models. The situation is a bit easier in a sense that the probe shanks are longer, so you can push out the fiber more easily further out there. So we don't have an adapter for that yet designed. We don't currently do that much things with the primates ourselves, so we don't have customer base for that. But if there are people who are interested in that, please contact me, and we can look at the longer probes separately later. So the basic idea there is that we hope to have the standard kit that are easy enough to use by any labs to get the fiber and the neuropixel shank in parallel, roughly 150 to 200 microns from each others, and that you can get let's say 6 millimeter deep in the brain with it. Then we had the next question.
So there were some practicalities, right? Like on how to glue it and how to paint it. So like the painting itself, I think that was the question first. That the painting itself, like you have to be very careful, and like I usually use a pipette tip, and only one drop of dye is coming out of it, and I kind of paint it like with the brush for 5 minutes until I feel that the probe accumulated a lot of these dyes. And during doing that, I'm very careful not to push down the probe to touch it with the glass, but actually having it contact, like to not contactless, to not to contact. And the other thing is actually from the glass, the dye is easier to solve down. So basically, when entering the brain, it is losing the most of the dye. How to align them? Like I did everything under stereo microscope with the scale under the fiber, and this is a 2-component glue that is getting rigid like in 15-20 minutes. So once you reached the perfect position, you just make sure that it is not moving, and you can leave it there.
Thank you. I think there were further gluing questions. So I think there are many styles in the different labs, and probably similar to Enter for most cases. And we try to make a useful adapter for that, and then hopefully it's useful. If not, labs will continue to do it on their own in any case. So I think we try to make that easier. Then there's a question here from Blake Russell. So
yeah, I'll take that one. So what is the minimum number of sites that the tapered fiber stimulates, which means how localized can you make the tapered fiber stimulation? So I'll share the screen very quickly to yes, I can use this one maybe. So as you see here, there is an illuminating region which is a bit more there than 500 micrometers for these 2 millimeter fibers. These, let's call it sub-active region, let me call this like that. Its length depends on the total length of the taper. For longer taper, it will be slightly longer. For shorter taper, we can go down to 350, 400 micrometer for the 1 millimeter taper. So it's a bit of a trade-off on how long you want to have the span for the light to be scanned and how localized you want to be. So let's say if 400 to 600 micrometer, depending on the total length of the taper.
Thank you. There's a question on comparison between the taper fiber and the micro LED based neuroprobe. So, Marco, do you have comment
yeah,
on that
so
entry?
I don't have personal experience with a micro LED based neuroprobe. The main thing that arises to me is that the fibers is inherently a passive device. So you can couple multiple wavelengths to the fiber. You can also collect light from the brain through the same device. So, and this is something that micro LED neuroprobe cannot do. On the other hand, micro LED, it may be just easier switching between position because you just turn on and off LEDs. These, I think, are the main differences between the two systems. And as the simulation will remain spatially restricted as you move it down the fiber by increasing the light injection angle? Yes, as I said, the length of the subsite moving along the taper is pretty much constant for the whole scanning span. You have just at the very end of the numerical aperture, but just because the fiber is going outside its working properties, and this length of the subsite reducing a bit, but then the power just get cut off from going outside the numerical aperture, and you won't notice in a real life application.
Thank you. There's a question of simultaneous EFIS and photometry in the same region. So I think it was an interest future outlook, but Marco, do you have examples of that or comments on that?
Oh, so I didn't really perform simultaneous experiment with the two techniques, so I'm not 100% sure, but providing that you're using the right wavelengths not to interfere between the sensors and the actuators. Besides photoelectric artifacts, I cannot see interaction between the two. Andrew, I don't know if you have some insight on it, or
I can only also just speculate. So in that case, like I didn't get it from the question whether it's EFIS plus photometry, so it's not optogenetics included. So in this sense, maybe I would put the fiber a little bit further from the probe to decrease the photovoltaic artifact, because that could be very strong when you are imaging basically with the fiber photometry. But I also only can speculate here.
Thank you.
And regarding the minimum distance possible between the fiber and the chunk of electrodes, this is really a mechanical limitation, mainly given by the radius of curvature of the fiber and how the neuropixels is made. So it will be challenging going below 100 micrometer, 150 micrometer. It will be very challenging due to mechanical constraint of the fiber and the neuropixel body itself.
Yeah.
Are there differences in light diffusion using lasers versus LED light source with the tapered fibers? So LED works fine for the full NA operation where you illuminate the whole extent of the taper. You just cannot get LED to work for the light scanning application, just because it's so difficult to properly collimate and focus the LED light for the nature of the emitter. So LED is a good way to go for the full taper operation, for sure, not so much for the scanning.
Yeah, thank you. I think we've covered all the questions I can see in the chat window. So I think maybe we wrap up now. To wrap up, I would like to thank Marco and Endre for great presentation. I have to say this was a really interesting one, and we had a big audience also, which sort of highlights that one. We will share the recording of this webinar in a week or two after we, get it to the right format at our website, so we can share the link for that. So it's also good for those who couldn't join today to see it. And as mentioned, we'll have the holders at our Sensapex SFN booth. So if you are there, please come by. You will see how the tapered fiber looks like together with the NeuroPixel in one example. And then if you have any further questions after you think about this webinar for a day or two, just send them over the email to me. I can pass them to Marco Entry or others who might have answers for those. So with this, I'd like to thank everyone and close the webinar for today. Thank you. Bye-bye.